Good morning, everyone. I, Dr. Poonam Joshi, welcome you all in our Saturday ACTRAC academic program. Today's presentation is by Dr. Sanket Shah. He is a senior research fellow, PhD, in one of the labs under Dr. Sanjay Gupta at ACTRAC Tata Memorial Hospital. And he will be talking on real-time PCR and its implication in cancer biology. So I welcome you, Dr. Sanket. You can start your presentation.
Thank you, Dr. Poonam, for the nice introduction. I hope my voice is audible.
Yes, you are audible.
Okay, and the slides are visible.
Yes, yes.
Okay, thank you. So let's start with the today's presentation. So today I'm going to talk about real-time PCR and implication in cancer biology. So first, let us know that what is real-time PCR and what is it used for. So when we say real-time PCR, it is connected with the PCR. So we know about the PCR, it is a polymerase chain reaction, amplification of the DNA. So even though you have a small amount of DNA, you can amplify it to a larger amount. Now the difference between PCR and real-time PCR is that in case of PCR, at the end, you get to see that what is the difference. And in case of real-time PCR, you can see in a real-time manner. So say for an example, there are two people, one is thin and one is healthier. So just looking at them without having their weight, we can say that one is healthier than the other. So that is PCR. But if you want to say it accurately, say the one person is having 50 kg and the other is having 75 kg. So if we have these numbers, then we can say that the other person is 1.5 times healthier than the first person. So that is real-time PCR. You can statistically accurately say that how much difference is there compared to the other one. So now, another beauty of this technique is that even if a single copy or half a copy is there, you can amplify it and you can detect it because the machine is very, very sensitive. We have just covered the difference between PCR and real-time PCR. Now, what is the implication? Of course, we are working in the Cancer Biology Institute, and most of the time we use this technique to detect the expression of gene. So we know that in case of normal cell, the expression of certain genes is not there. In case of cancer, the expression of certain genes alters. Some of the genes expresses at a higher rate, and few of them expresses at a very low rate. So to detect their expression, we extensively use this technique. Second, for the viral quantification, and the others are mentioned over here, like food testing, animal and plant breeding, and forensic. So for an example, in case of cancer, how can we use this technique? We all are aware about the BRCA1 gene, which is often mutated in case of triple negative breast cancer. So what we do is we design a specific primer and then we look at the expression of BRCA1. And while look at the expression of BRCA1, you can just say that what is the alteration in case of expression. So it might happen that some of the patients are having higher expression of BRCA1, some of them are having mutation. So everything, say mutation or the higher or lower expression can be detected through real-time PCR. Secondly, like just connecting with the cancer. So if the radiotherapy or chemotherapy treatment is being given, then one can monitor the expression of particular gene, and it can be used as a prognostic marker. And also in case of other diseases, say viral infection is there, and if the treatment is ongoing, then how much viral load is there? Like just a live example for the COVID-19, we get the CT value. So that CT value shows us the load of virus in our body. Lower the CT value, higher the load. Higher the CT value, lower the load. That I'll come to. So let us start with how does this real-time PCR work? Okay. So first we need to understand the basics of polymerase chain reaction. Now here you can see a DNA, double-stranded DNA. One of the strand is 3' to 5', that is red color, and the second strand black color, which is 5' to 3'. Now say this is a single copy. So we are starting with a single molecule of DNA. We put these nucleotides, primers, and enzyme, and we add all of them in a single tube. So what happens when you put all of them in the same tube? So first, we set up a protocol for denaturation. So because of the denaturation, both the strands will be separated. So we can see here, one strand is here and the second strand is here, and these are the primers. So in the second step, we put a temperature of around 60 degrees Celsius, which is efficient for the binding of primer. So at the second step, annealing of primers to the template DNA takes place. Now comes the extension step. So in the third step, the enzyme, which is known as Taq polymerase, that binds at the 3' end and extend the strand. So further, like additional nucleotide will be added and the new strand will be complemented. So now we can see that we started with a one molecule, and now at the end of one cycle, we have two molecules. So this is a calculation with the power formula of 2 raised to power n, where n is the number of cycles. So after one cycle, from 1 to 2 molecules will be there. After 2 cycles, from 2, 4 molecules will be there. After 3 cycles, from 4, 8 molecules will be there, and likewise. So if we are talking about say 5 cycles, then it will be 2 raised to power 5. So 32 molecules will be there. If we are talking about 30 cycles, then it will be 2 raised to power 30. Whatever number will come, those many molecules will be there. So you can understand that from a single molecule, we are now having 2 raised to power 30 molecules, which is quite a good amount for the detection. So yeah, so this is like after one cycle, after 2 cycles, and so on. So this is how the reaction mixture looks like. And we put real-time PCR for 30 or 40 cycles. And this is how the reaction looks like at cycle 25. Now one might ask that if we are, what if we are starting with 10 number of molecules or say 100 number of molecules? Will it get exponentially amplified? So now let us see this. So for an example, at cycle number 25, we are having a soup of all the things, nucleotides, primers, the amplicon, everything, because these all things we have put in the tube. And at the end of 25 cycle, we have 10 lakh molecules. So just as per our calculation, what will be the number of molecules at cycle number 24, 23, and 22? we know that the amount gets doubled. So at cycle 24, it will be half, say 5 lakh. At cycle 23, it will be the 2,50,000. And at cycle 22, it will be 1,25,000. So this is how the number increases. So this is how the graph looks like, where one can see that on the x-axis, the cycle number is there, and on the y-axis, the number of molecules or fluorescence is there. So at cycle 25, 10 lakhs molecule is there. So now the question is, what will happen at the 26th cycle? So here on the scale, you can see that 20 lakh is there. So it will reach up to 20 lakhs. Now the question is, what will happen at cycle number 27? Well, as per the standard statistical calculation, it should go to 40 lakhs from 20 lakhs. But this is not what happens, because this reaction has a limitation. So just I have said that if we start with the 100 molecules, it does not happen that after 30 cycles, each and every molecule will be amplified. So there is a saturation, because we put at a particular concentration everything. So we put primers, we put nucleotides, and everything has a specific concentration. So the purpose of putting specific concentration is that the reaction should not get saturated. And we should just have 20 lakh or 40 lakhs molecules for the detection. That much is enough for us. So what happens once it reach to particular range, say here in case of 45 lakhs, you can see that after cycle, say 27 or 28, the graph is getting saturated. So we are having a step increase followed by the straight line or the plateau line. So that is what happens. So if you start with a very high amount, say 1,000 molecules in one case and 10,000 molecules in another case, so after 30 cycles, both will get saturated, and the difference will not be observed in the normal PCR technique, but the difference will be observed in case of real-time PCR technique. Why? Because we put up a threshold. So say here, the 10 lakh range is there. So that will be our threshold. So we put up a line over there. The machine itself does that, and that will be our range. So here is the example. Just a second, the slide is not moving. Okay,
Sanket,
fine. I think, yeah, I can just cover it. So the thing is, let me give you an example. So the range is 10 lakh over here. So that is the standard or threshold range we put for the reaction. So say you have the cut margin sample, and second sample you are having is a tumor sample, and the third sample you are having is the treatment sample, any kind of treatment. So what happens is each and every sample is having different amount of starting amount. So in case of cut margin, say the CT value is around 28, and in case of cancer, the CT value is 25, and in case of treatment, the CT value is 23. So now what does this number represent? So 28, it means that to reach the threshold, that is 10 lakh, it takes 28 cycles to reach over there. In case of cancer, we have just said that the 25 number, it means that to reach up to 10 lakh range, it needs 25 cycles to reach over there. And in case of treatment, it needs just 23 cycles. So this is an inverse correlation. The lesser cycles required to reach a particular number, the higher expression of a molecule is there. So now what can we say that from 20 to 25 to 23, how much of difference in terms of expression is there? So one cycle, we know that after every cycle, there are two molecules which are getting amplified or which are getting doubled. So the difference between 28 to 25 is 3 cycles. So as per the standard calculation, the difference will be 2 raised to power 3. So 2 raised to power 3 will be 8-fold change. So we can just have a statistical calculation that compared to the cut margin sample, in case of cancer, there are 8 number of molecules which are higher because the CT value difference is 28 and 25. So 2 raised to power 3 will be 8-fold change. Now what will happen in case of treatment where the cycle is 23? So again, the difference will be one is 28 and the other is 23. So again, the differences will be 5 cycles. And how the calculation will be? It will be 2 raised to power 5. So that is 32. So 32-fold change difference will be there. And we can say that in case of cut margin, when there is a single copy, in case of cancer, there are 8 copies. And after giving treatment, there are 32 molecules or 32 copies. So this is how the data is basically used in clinics, where we can just directly correlate with the number of copies. So yeah, that is about the standard real-time PCR. Now, one may ask that is it a straightforward calculation where we can directly say that based on the cycle number, the expression is more or less? Well, the answer is no, because it might happen that you are putting more of DNA in case of the cancer sample, and you might get more expression. So what happens is we take a standard control, which is also known as housekeeping gene or internal control, and we check the expression of that.
Dr. Sanket, sorry for interruption,
yes.
Dr. Poonam here. Can you upload your slides again, if possible? I think it's not moving. Yeah,
yes,
because that would be
right,
better for us to correlate and understand as clinicians. So I think you can, you need to upload it again. Or you can go to the PowerPoint mode and see. Yeah, please upload it again.
Yes, just a second. Okay, great. Can you see this slide, the Excel sheet?
Yes, started screen sharing, not yet, but yes, now we can see the Excel sheet actually right now.
Yeah, I have uploaded that one. Okay.
Yes, we can see.
Okay. Wonderful. Okay. The slide is visible, right?
Yeah. Please go ahead. Yeah.
Okay. So this is what I was talking about in case of cut margin, in case of tumor, and in case of tumor plus treatment sample. So this is the standard threshold where the blue line everyone can see. So this is the blue line which is put by a machine. Okay, and this is the range where the wherever the line will reach over here, it will be considered as a standard CT value. So this is one sample, this is second sample, and this is third sample. all of them are reaching at this range at a different cycle number. So this is the cycle number. In case of cut margin sample, this is cycle number 28. In case of tumor sample, this is cycle number 25. And in case of tumor plus treatment, this is cycle number 23. So we can see that from 28 to 25 to 23, there is a difference of 3 CT value, and further the difference of 2 CT value is there. And how do we calculate? So the formula is 2 raised to power cycle number. So here again, 3 cycle difference is there. So 2 raised to power 3. It means that 8-fold change more expression is there in case of tumor. Again, in case of tumor plus treatment, it is 23. So from the cut margin sample, the difference of 5 CT value is there. It means that 32-fold change higher expression is there after giving the treatment. So now, the thing is, like I said, that in case of standard calculation, we just cannot directly correlate with the.
Hi, Dr. Sanket. I think
Yeah.
again that, sorry, again, we are not, yeah, please share.
Yes. Actually, I'm sharing the Excel sheet. I hope it is visible.
Yeah, the Excel sheet is visible. Yeah.
Yes. So this is the same example, say 28, 25, and 23. But we need to look at the internal control gene. Okay, so this is the internal control, which is RPS first. So in case of RPS, you can see that in case of cut margin sample, the value is 18. In case of tumor, the value is again 18. And in case of treatment, the value is 18. So what does this represent? This represents that you have put an equal amount of DNA in all the 3 tubes. Okay, because this might just happen that you have put more amount of DNA in case of tumor sample, and that is why you are getting lesser CT value. So this we need as a confirmation that amount of DNA is equal in all the samples, and then you can proceed with the further calculation. Now taking the second example, okay, in the green color, 28, 25, and 23, those CT values are same, but now look at these values of internal control genes. So here the internal control is RPS 13, which is ribosomal protein subunit. So in case of cut margin, the value is 18. In case of tumor, the value is 15. And in case of treatment, the value is 13. So we know that, again, in case of tumor, there is more amount of DNA, which is 8-fold, and the difference is same over here. And in case of treatment, the difference of 5 CT value is there for the internal control gene. So we can't really just say that the higher expression in case of tumor is there for this 25 and 23 CT value. So this is how we do the calculation. So first we calculate a delta CT, that is a difference between internal control and the standard gene. So gene A is our target gene, and this 18 is the value of internal control. So difference between 28 to 18, that is 10. And here the difference between 25 to 18, that is 7. So now we calculate delta CT. So delta CT is just a reference. So reference means you need to take one as a reference. When you say that it is upregulated in case of cancer, you need to have some reference, which is a cut margin sample, which will be considered as a 1. So this will be like 10 minus 10. So that will be 0. And here again, from 7 minus 10, so it will be minus 3. And again, when you calculate the change, it is 2 raised to power 0 is equal to 1. And here 2 raised to power minus 3 will be 8. So this is how we calculate. So I have just said that it is 2 raised to power n. So that is 2 raised to power 3. That is 8 difference is there. But first we subtract it from the internal control gene, that is 25 minus 18. And then we proceed with the calculation. So here we were talking about the second example where the value of internal control gene itself is changing. So when you calculate the difference, there is actually no difference. In all the 3 cases, there is equal amount of DNA. So we just can't say that the higher expression is there. I hope everyone understood this example, or you want me to go through it again?
I think we'll go ahead with the rest of the presentation, Dr. Sanket.
Okay, fine. Okay, so now let us start with the next part, that how much sensitive this technique is. So I said at the beginning that even though a single copy of DNA is there, then also we can detect the expression. So here the example is like obviously you guys can understand the tissue and blood more. So even though 200th or 150th of microliter of blood is there, then that much amount is enough to isolate the DNA or isolate the RNA, and then we can proceed for the technique. Yeah. So how do we detect or measure the DNA? So there are agents which are conjugated with the fluorophore, and they give the fluorescence. They bind to the double-stranded DNA and they give the fluorescence. So one example is ethylium bromide. This is an intergalactic dye which binds to the double-stranded DNA. And the second example is cyber green one dye. So cyber green has a fluorescence and it binds to the DNA. So you can see here that when the DNA is single-stranded, the cyber green cannot bind. But as soon as the DNA is getting amplified or it is getting double-stranded, the cyber green molecules are binding. So the more number of DNA or more number of copies will be there, the more amount of cyber green binding will be there. And as a result, there will be emission of more amount of fluorescence. And that is what we capture. So more the amount of fluorescence, more the amount of DNA. So what about the instrument? So there are three parts. One is thermal cycler, the second is optical module, and third is the computer. So this is how the machine looks like. So this is the thermal cycler where we set up the protocol, denaturation, annealing, and extension. And this is the optical module. So we remove the optical module, we put up a plate over here, and then the fluorescence will be captured from the LED array. So this is how machine looks from the inside. So whatever reaction is there in the plate, the fluorescence will be captured by LED array and the result will be displayed on the computer screen. So yeah, that is about the real-time PCR. I would like to take any question. If it is there, would love to take the questions.
So Dr. Sanket, I think, could you again please explain to us the difference between the PCR and the real-time PCR? I could understand a bit, but I think that's the very basic which we should understand because the whole topic is on that only. Yeah.
Yes, yes. So the main difference between PCR and the real-time PCR is that in case of PCR, you put up a reaction for 30 cycles. So after 30 cycles, whatever difference will be there, you'll be able to see that. So if you are starting with a very higher amount of DNA, and like we have seen in the case of graph that it gets saturated after a certain point. So after say 25 cycles, both the reactions will get saturated and you won't be able to see the difference. Whereas if you will do the same thing in case of real-time PCR, because you are setting up a threshold at a very early stage, you will be able to see the difference. So now let me just give you an example. That like the example which I have given, there are two people in front of us and we don't know their weight. Okay, so we can just say that one person is thin and the other person is healthy. But if you have real-time PCR, then you can accurately say that how much difference is there. How much difference is there? That is point number one. And point number two is, even though like the people who are very much similar in terms of weight, say one is 90 and the other is 92. So from visually, we will be able to say that okay, both of them look similar. There is hardly any difference. But if you do a real-time PCR, then the difference of 2 kg will come. So it will come in the picture. So that is the main difference between the conventional PCR and the real-time PCR. That one is exact how much difference is there in terms of fold change. That okay, this much times higher DNA or this much times lower expression is there in case of cancer. And the second difference is even though visually it appears similar, but actually there is a minor difference or minute difference which you will be able to capture through real-time PCR.
Thank you, Sanket. I think there's one more question. I could not understand it well. What is the clinical application of some genes whose expression is decreasing in cancer? Can you please explain, Tirtha, what exactly is the question?
Yes, yes, yes. So, like we have taken the example of gene where the expression is increasing, say from 28 to 25 CT value is there. So 25 CT value, it means that the expression is more because the relation is inverse. Now, if we take the vice versa example, say in case of cut margin, the expression is 28, and in case of cancer, the expression is 31 or 32. It means that it needs 31 or 32 cycles to reach over there. It means that the expression is there. So now, obviously, when in case of cancer, the expression of one gene is going down, then you cannot target it because the expression is going down. But you can use it as a prognostic marker. So you can just use it as a predictive marker that, okay, what if the expression of this gene goes down? So you can just take the sample, you can check the expression if it is a standard marker, and you can use it as a prognosis, both in case of treatment and also in case of cancer.
So any other question anyone would like to discuss with Dr. Sanket? So I think with this, we conclude today's session. I thank Dr. Sanket for making relatively a difficult topic easy for us, because as a clinician, it's a bit out of our league. So I think thank you so much, and we would request you in future also to take our more classes on the basic science for cancer. Thank you, Dr. Sanket.
Sure, Dr. Poonam. If any question is there, one can reach out to me. I would be happy to discuss.
Okay. Thank you, Dr. Sanket.
Okay.
I think
Thank
there are
you.
no more
Thank
questions.
You.
Thank you. We will conclude. Thank you so much.
It has been of 10 days. Okay. Okay. Okay.